Hello, everybody, and welcome to this first module in our online MOOC about vector-borne diseases under climate change. My name is Anna-Sophie Stensgaard, and I'm a professor at the University of Copenhagen in Denmark, and I'm also the coordinator of the project called Prepare for VBD, which stands for a cross-disciplinary alliance to identify, predict, and prepare for emerging vector-borne diseases. This is the first module in our MOOC, and here I will give you a broad introduction to what vector-borne diseases are and how they are impacted by climate and environmental changes. And also, of course, briefly to our project and our consortium. So what are vector-borne diseases? Vector-borne diseases are illnesses that are caused by a variety of pathogens like parasites, bacteria, or viruses that are transmitted to humans and animals through the bite of infected arthropods, such as mosquitoes, ticks, and, for instance, fleas. These vectors consist of a very diverse group of living organisms, typically insects, but it can also be other invertebrates, for instance, snails and ticks. And they can transmit these pathogens from human to animal or animal to human or between human to human, as illustrated here in this figure. Vector-borne diseases is a growing problem. It's already accounting for about 17% of all known infectious diseases. And together they are affecting more than 1 billion people globally every year, leading to more than 1 million deaths. Many of them are also zoonotic. We cannot isolate them really as a human or an animal health issue. And the problem is growing. They are continuously making headlines because they are on the move. So we are, for instance, seeing increased outbreaks of Rift Valley fever in both Africa and Europe, dengue fever, West Nile, chikungunya, fascioliasis, schistosomiasis, and so on. So why are we seeing these changing patterns? Why are they continuously making these headlines? First and foremost, it's important to understand that these vectors and the diseases that they can transmit, they do not exist in isolation. They're all part of the same ecosystem. And that means when we, the humans, are causing climate change or land use changes or the biodiversity crisis, we will impact the health of both humans and animals because these are very closely intertwined, as illustrated here in this triangle. It goes for many diseases and for health in general. However, vector-borne diseases are particularly sensitive to climatic and environmental changes because they have or need this vector, this invertebrate, that's very sensitive to changes in temperature, precipitation, or in general to the habitat in which they are existing. And that is part of the explanation for why we are seeing these changing patterns, because climate change, as I'm sure you know, is real and it's ongoing. We know that the temperature has already risen about 1.2 degrees Celsius since the late 19th century. And most warming have occurred in the past 35 to 40 years. And this means that for many vector-borne diseases or vectors, globally, the climatic suitability for these are increasing as many of them lack warm or tropical conditions. This is illustrated here, for instance, in one of the Lance's countdown reports. You can see the graphs here for dengue, for Aedes mosquitoes that transmit dengue, and for the mosquitoes that transmit malaria. The changing climatic conditions are increasing these suitable areas, especially for the mosquito-borne diseases. And in the past 60 years, for instance, the global climate suitability for the transmission of dengue caused by Aedes mosquitoes have increased about 15%. Likewise, we see that the climatic suitability for malaria transmission, for instance, in highland areas, the cooler areas in the African region, has increased by almost 40%. Many vectors are indeed expanding their geographical range. And here you can see some examples of some vector species that have increased distributions or changed their phenology, which means potentially having a longer transmission season due to, amongst other things, increases in global temperature. It could, for instance, be biting mixtures transmitting blue tongue virus, tick species that can transmit Lyme, TBE, babesiosis, Q fever, etc. And of course, also mosquitoes, especially a mosquito such as Aedes albopictus, that, or known as tiger mosquito, that can transmit dengue and chikungunya and Rift Valley, and also Culex mosquitoes transmitting, for instance, West Nile fever and avian malaria. The challenge that we're facing globally is that these diseases and their vectors do not really respect any borders. And we need to know which diseases or vectors are going to occur where and when so that we can be better prepared. And to do so, there is a need to improve our surveillance and also our capacity to discover and detect these diseases at an early stage. It is a global problem, this. It's cross-cutting, it requires an interdisciplinary approach, and it also requires that we both have depth and breadth in the knowledge about this problem. Unfortunately, we do tend to operate in so-called silos, both across disciplines scientifically, also across sectors, but also how we work with, for instance, the different vector groups, as illustrated here in these three pillars, or you could also call tooths of a comb, representing mosquitoes, ticks, and snails, for instance. What we need is a more comb-like approach, where we also acquire some breadth in our knowledge, that we learn a bit from each other, and the depth and the experts that we have in each of individual fields. And this is what we're trying to do in the Prepare for VPG project. So we are a consortium consisting of 10 complementary research and policy institutions in Africa and Europe. And we, as you can see on this map here, cover a vast geographical gradient of ecosystems and climates in which our target diseases and vectors are distributed. Our overall aims and our target organisms and diseases is first and foremost to create a broad conceptual knowledge about vector-borne disease emergence, how they respond to climate change, and something that reaches beyond specific disease or specific vector or specific country or specific scientific discipline. With this, we aim to improve how we can project and prepare in both Africa and Europe for a number of zoonotic vector-borne diseases. In particular, we are focusing on the illustrated here three groups of vectors. The ticks that can transmit anaplasmosis, theileriosis, and babesiosis. A group of freshwater snails that can act as intermediate hosts or vectors of a number of different snail-borne diseases, parasites. And finally, mosquitoes and mosquito-borne diseases represented in our project with Rift Valley fever. How are we going about this? Let me illustrate this using this figure here, which is adopted from invasion biology, where you can see time on the x-axis, and then on the y-axis you can see you could say the abundance or the number of infected cases of an emerging vector-borne disease, and also the associated health and socioeconomic costs with actually being invaded by a new vector or disease. So usually what many countries are doing is a sort of reactive approach to this. We do not react until we detect the disease, for instance, in the human host or in our livestock, at a time where we might already have a lot of vectors and a lot of cases already. So that means prevention and eradication becomes highly unlikely and certainly also very costly. So what we suggest to do, or our approach in our project, our consortium, is a bit more proactive. So we will focus, or we did focus, on the environmental phase to increase the knowledge we have about vector ecology, to develop new and sensitive detection tools and model-based surveillance to increase early warning capacity. And as I mentioned, we are working cross-organismal. We're casting the net wide, trying to address a quite diverse group of vectors and zoonotic vector-borne diseases to enable this cross-organismal learning and more integrated approaches to surveillance, prevention, and control. So in our project, we've got a number of different work packages, and some of the other modules in this MOOC will cover these themes. Very overall, they are all organized within 4 overall themes in the project. And each individual partner in the project has been working on this and will present to you their findings in the following modules. What's important to take home from this is that we have all worked together to do this, and we have all worked across this vast geographical and climatic gradient, ranging all the way from South Africa to Denmark, to be able to ensure that we actually are studying any potential variation in the responses of our vectors and their diseases across this very vast gradient. So let me just also bring your attention to some of our outreach material and training material. We have a dedicated YouTube channel where you can go in and you can see some of our training videos that we have developed, not these MOOCs, but other training videos. And then, of course, also we have a dedicated website, prepareforvbd.eu, where you can find our newsletters, all the publications that we've been making, info packs, field guides, student blog posts, where you can see events that might occur, risk maps that we have been developed, and a lot of other different resources. So please go and visit our website as well. So with that, our of course would like to thank you very much for tuning in for this first module in our MOOC about vector-borne diseases in the context of the Prepare for VBD project and consortium. I hope that you found it interesting, and I also hope that you will tune in and watch the reminding modules and find those interesting as well. So thank you very much for your time.
In this module, I will teach you about how to model vector-borne diseases under climate change with the case of Fasciola species. So, climate change already influences the ranges of a lot of species, and a very classic example is if you look at a mountain. So, if you think of a mountain, you can think from the top, maybe all the way at the top of a mountain, there's just snow and ice, and there aren't any species at all. But as you go down the mountain, you might start to see grass, moss, and then you might see trees and agriculture further down. And with climate change, what we've seen, you can see here on this picture. So these researchers, they went up the mountain and recorded exactly what kind of species they were finding in what stages of the mountain. And they compared this to how it looked like 200 years ago. And you can see these range shifts. So trees and vegetation going further and further up the mountain. And this is because of climate change. And the reason this is that, well, these trees, they need certain conditions, and you will only find these below a certain elevation. And we will also find a lot of species that are shifting towards the poles, so further north or further south, away from the equator. So you might think of each species as having some range of, for example, temperatures that it can tolerate. So there's some perfect temperature where a species, it can move very fast or grow very fast. But if it gets too cold or too hot, a species might not be able to move very much or reproduce very much. And if it gets way too cold or way too hot, at some point, an organism just dies. And this we will call a niche of a species. So the set of, for example, temperatures that a species can tolerate, a population can grow and reproduce, that we will call the niche of a species. And so in the mountain, with the temperatures changing, all of a sudden areas further upslope on the mountain, they're within the niche of trees, and that's why after these 200 years of climate change, all of a sudden we find them there where we didn't before. So with Fasciola, it's extra interesting because this is a parasitic species, and here you can see the life cycle, which we'll think about a lot in this module. And in the life cycle, of course, there is the mammalian host of a fasciola, so cattle or sheep or humans, but also a snail host. And then there's the free living stages. So if you think about this kind of curve where conditions need to be within a certain range so that a species can survive, with fasciola, it not only has to be within the range of the parasite, but also the snail species and the mammal host has to be able to survive. With fasciola, there are also two different species. So one of them is Fasciola hepatica, and one of them is Fasciola gigantica, and they have very similar, or they have kind of identical life cycles, similar strategies, but they use different snail hosts, and they're also found in different areas. So Fasciola hepatica, a very common host, at least in Europe and also in Africa, is a snail that's called Garbotroncatula. And for Fasciola gigantica, it's a different snail, for example, Radix netzelensis. So here we're asking, well, we know kind of the historical distribution, at least approximately, for these two species. So that you see on the left in gray, Fasciola hepatica, we find it all across Europe, all across the Americas, also in Australia. And then Fasciola gigantica, we find it in the tropics. So across Africa, also in South Asia. And there's a little bit of overlap also. And now we're asking, just like with the mountains where we see these species go further up the slope, well, with climate change, how will this distribution change? And of course, one way we could start thinking about it is to look at where is the species now, right? So we have records for all kinds of species. We know where they are, and then we can measure what kind of environmental conditions, so temperature, precipitation, are there in those areas where these species occur. And then we could think that in other places that have similar environmental conditions, well, that's where the species might also occur, or in the future, that's where the species might migrate towards. So if we're going with this strategy, and we call it correlative modeling, it always starts in the field. So here are some photos of field trips with Prepared-MOOC project. So it always starts with people scooping snails in the aquatic vegetation, identifying the snails. And luckily, a lot of researchers have done this for a lot of time, and they've published this data or uploaded it so we can collect all of this data. And here you can see for the two species I mentioned at the beginning, so Radix netzelensis and Garbotroncatula, in red and in blue, where these species have been recorded. So you can see that Radix netzelensis has been recorded in many areas in Africa, and Garbotroncatula, a lot of occurrences in Europe, and also a few in Africa, for example, there in South Africa, and also in the East African highlands and in North Africa. So here's those same occurrences, but now I've plotted them together with the temperature and the precipitation in these areas. And already you can see a few patterns. So in Europe, it's much colder than in most areas in Africa. And in the few areas in Africa where Garbotroncatula occurs, also it's relatively cold. So in Morocco, in the highlands, and in South Africa. So that's on the left. And then on the right, you see the precipitation, and you'll see that in areas with very low precipitation in yellow, there are almost no occurrences of any of the species. So here we can see already, or we can start thinking about what kind of environmental conditions these species will need. So here again is those same occurrences, but now instead of only in geographical space, so on a map, I've plotted them where you can see the temperature and the precipitation at the places where they occur. So there's the average temperature over many years. And you can see very clearly that these two are separated. So Radix netzelensis, it needs high temperatures above around 15 degrees, but then Garbotroncatula will find it even in places where it's below zero. And also all of these, you see they are kind of up. There are very few occurrences in places where there's little precipitation. So this data we can feed into models, and they will make these correlative assumptions. Now I've simplified it a little bit with just annual temperature and annual precipitation, but you can think that there are many other ways we can put this, or variables we can use. For example, what is the temperature the hottest month, or You can use satellite imagery and so on. And then we can get pictures like this, where this is the model's prediction of where you are likely to find these species, not based on the places where they are found, but based on the environmental conditions at these places where we find them. And because we did it with the environmental conditions of where we find them, we can then take predictions for what the climate will look like in the future, and we can then project towards those climates also. And that's what you see in this picture. So again, those same species, but under current climatic conditions, and what we predict, what we expect that this will look like in the future. So red colors means it's very suitable, you're likely to find a species there, and blue colors means you're unlikely to find a species there. And here you can see quite clearly that, at least for Galbert truncatula, you're less and less likely to see that species both in Africa and in Europe. So the conditions, they are moving away from the conditions that are similar to those where we typically find Galbert truncatula. And that's probably because, as we saw earlier, that you find it often in places where it's cold and where it's wet, and the planet is warming up with climate change. So just to recap, we've done this exercise in correlative modeling very quickly, where we saw that you go from occurrence data gathered in the field, combine those with environmental data, so temperature, precipitation, and we combine those with machine learning, with statistical models to make these suitability maps, where you're likely to find a species, in this case, intermediate host for Fasciola. Now I'll take a step back and look at a totally different way you could do this. Because of course, maybe you thought when I showed this idea of a curve, in the middle of the curve and the temperature is perfect, the organism is performing very well. Well, maybe you thought, can't you measure this in the lab? I mean, we can catch these snails. I'm sure we can measure how fast they grow, and of course you can. So this is a totally different way of doing this. Is saying, okay, with experiments in the laboratory, we can actually measure what kind of temperatures these organisms prefer. In this case, we'll look at what kind of temperatures does actually the parasite prefer. So again, on the right is the transmission cycle, and you can see there are many stages where the parasite is, well, outside, exposed to other weather that it experiences. So already after excretion, these eggs, well, they need to hatch. They do that just in the water. Then they need to get inside the snail. They need to develop inside the snail. And then they need to swim and insist on the vegetation. So all of these are traits, and we can have these parasites in the lab, and we can measure them. So this is, I mean, some photos of some experiments we did here in Copenhagen in the Prepare project. Just have these snails increase the temperature, and you measure how they behave. Well, this also researchers have been doing for many years, and we have data from all of those years. So, for example, in this graph, you'll see for around 10 different experiments, 10 different publications done by different people at different times and different places, and they've measured all the same thing, which is how long does it take for a Fasciola hepatica egg to hatch. So this is the egg that's excreted from the mammal host. How long does it take for that one to hatch and become a merocedia? And this is plotted here on the left, so the temperature on one axis and the number of days that they measured on the other axis. And you can see this very clear pattern, even though, again, this is different people that did this at different points in time. But the pattern is very clear, and of course we can make a curve through this that will look something like this. And then again, we can predict, okay, at this temperature, it will take so many days for an egg to hatch. And you can see if it gets too low the temperature, below 10 degrees, well, it takes a very, very long time to hatch. And so you might think, well, this is kind of below where the parasite still can still can reproduce and be transmitted. So here is the same thing, but for all different traits, representing the different parts of the life cycle. So just go through it from the top left. There's the hatching time that we just saw, and in red is for Fasciola gigantica, so the tropical species, Fasciola species, and in blue for hepatica, so the temperate one. you can see this clear difference. I'm not showing all the points from the different experiments here because there will be a lot of colors, but all of these are based on this experimental data. Then here on the second one is the pre-patent period, so how long it takes for a snail that is infected to start shedding the Saccharine. Then the next one is the infection efficiency, so how often actually this fasciola is successful in infecting a snail. Then Saccharine release, so how many Saccharine does one snail release? And you can see also here there's a very clear connection. If it gets too hot, the snail is suffering so much, or maybe it's dying before the parasite can mature, and so it's not releasing a lot of Saccharine. And the other one is hatching success of the egg. So at 40 degrees, sure, the hatching time will be very low. The egg will develop very fast, but actually almost all of the eggs, they will die from heat before hatching. And then the last one is the total transmission strength. So combining these 5, and then we can predict how at different temperatures how able is the fasciola to complete its whole life cycle. And you can see for the 2 species a big difference. So the hepatica, from around 10 degrees, it can start completing its life cycle. At 18 degrees, it's just perfect. And at around 30 degrees, it cannot complete a life cycle anymore. And for Fasciola gigantica, it's from around 15 degrees, peaking at 25, and until around 35. And of course, this, combined with environmental data, temperature data again, we can put on a map. So we can say how close to those perfect temperatures is it throughout the year in different places. So here again, it's in blue. In blue, it means it's never within the suitable range, and in red, it means it's kind of perfect all year. And this, of course, again, with the model outputs for future climates, we can predict what would this look like under future conditions. And that's what you see here for both Fasciola hepatica and Fasciola gigantica, for current conditions and future conditions, seeing how this changes. And you can see that for both, at least in Africa, for both, the conditions are becoming less suitable. So too hot under future conditions is getting too hot for Fasciola species to efficiently complete their life cycle, at least as efficiently as now. In Europe, it's a little bit differently. There you can see that both species, they're becoming more able to complete their life cycle. So the colors are becoming more yellowish. So again, to sum up, here we did an exercise in mechanistic modeling. So here, other than with the cognitive modeling, we start with experimental data, measurements of how long it takes for eggs to hatch, for example, at different temperatures. Then we made curves out of this, thermal performance curves. And from these, we made suitability maps. So what I would like you to remember from this module is first this idea of an environmental niche. So every species in the world, including, of course, parasites and the hosts, they're constrained by what kind of environmental conditions they can tolerate. And that we call the environmental niche. Parasites, they're extra interesting because they have this complex life cycle. They're constrained not just by what they themselves can tolerate, but also what the host can tolerate, because they need those to complete the life cycle. Because of climate change, well, we can understand with this niche concept in the back of our minds, can understand that climate change causes range shifts. Because species, if new areas become within the niche, they will migrate there, they will appear there, and they will also go extinct, disappear from areas that become outside of their niche. To predict these shifts is not straightforward, but we have different types of models, different strategies to do this. One strategy is to use occurrence patterns, and we call those correlative models, so starting with occurrence data. And another strategy is started with experimental data. We call those mechanistic models. And I would finish by saying that none of these strategies are perfect. All of them have a lot of downsides. So we would try to do in this project, and I would recommend to anyone trying to make similar models, is to see these as complementary strategies. that can help each other, and in the end you'll have better models. So thank you very much for listening to this module.
Hello, my name is Joost Verhoeven, and I want to give a very brief introduction on the microbiome of ticks and why we study them in the context of vector-borne diseases. First of all, what exactly is a microbiome? While there are multiple definitions, and sometimes it can be a bit confusing what people mean when they mention the word microbiome, more often than not, they are referring to the complete collection of microbes, bacteria, archaea, fungi, that are associated with a certain environment or living creature. For example, let us look at humans. We carry with us millions of bacterial and fungal cells, and many parts of our bodies have a specific microbiome, from our skin to our intestines. These microbial partners are actually crucial for our health and intrinsically intertwined with our biology. As an example, our skin microbiome helps fight off foreign infections, and the bacterial cells in our gut play key roles in helping us efficiently digest our food. In the scientific world, and even amongst the general public, it has become clear that microbes play critical roles and exert a lot of influence in the world we live in. As such, it is not surprising that many studies are now focusing on understanding the microbiome of many environments. But how does one actually study it? How do we identify all these microscopic organisms in a specific niche? Often, this is done through the sequencing of a genomic material, such as DNA. If you have no experience with this, it may sound complicated, but at its simplest form, it means that we take a sample of a certain environment, extract the DNA from all the cells in that sample, and then sequence that DNA. In this context, that means analyzing what the composition of that DNA is, or what is the sequence of nucleotides, the building blocks that make up the DNA, for all the DNA present in the sample that we are investigating. This is useful because if we have these sequences, we can compare them to the reference database to assign a taxonomy to the sequence. That is to say, what is the sequence that we are finding? From this data, we can then reconstruct an overview of the community of microorganisms that was present in the original sample. Ticks are important vectors for disease in humans and other animals, and they also have microbiomes. However, unlike humans, their microbiomes remain fairly understudied and not fully understood. At the very least, it is believed that the microbiome of ticks play crucial roles in tick health and behavior, and is biologically important for ticks to be able to perform nutrient synthesis and digestion. In addition, it is also thought that during the process where ticks feed on blood, there may be important interactions between the tick microbiome and the host skin, facilitating the blood uptake and success of feeding. Evidence is also emerging that the microbiome of ticks is also related to their vector capacity. That is to say, the presence of certain microorganisms can have an impact on how competent a tick is in acquiring a pathogen and subsequently successfully transmitting it into a new host. This is of course interesting. The fact that the microbiomes of tick could play a big role in vector competence has large-scale implications. If we could improve our understanding on the interplay between ticks and their microbiomes in relation to their vector competence, we will likely reveal new insights and spawn new paradigms to control tick-borne disease. For example, perhaps modulating the microbiome of ticks could prevent the transmission of a pathogen, or a specific microbial interaction that is important for vector capacity can be identified as a target for a therapeutic solution. In addition, besides control and prevention, another valuable aspect of understanding the tick microbiome is that it could offer us a way to predict if specific pathogens are constrained to a certain locality or country. For example, hyaloma ticks spread the Crimean Congo hemorrhagic fever virus in a limited number of countries. But could ticks from other species also potentially transmit this virus? Does the microbiome play a role in this transmission? And thus, is there a risk of this virus spilling over into new ranges? If we could isolate the microbial players that are necessary for the transmission to occur, we could use this data to chart and predict future biorisks and mobilize associated public health investigations. For example, in our research within Prepare for VBD, we investigated the microbiome of over 400 ticks spanning 3 countries. In addition, we used specific tests to see which of these ticks were carrying pathogens with implication to human and animal health. By connecting this data, we were able to identify specific bacterial groups within the microbiome that were present at higher concentrations when one or more of the pathogens was also present. This could indicate that ticks need these bacteria to be able to take up and transmit the pathogens we detected, and that they could be a key aspect of vector capacity. Of course, this is just a very brief overview, but hopefully this illustrates that microbiomes play a huge role in many organisms, including ticks, and that studying this microbiome could give us valuable insight into the life cycle and vectorial capacity of ticks.
Hello everybody, this is Jörg Joris.
Hello, this is Thomas Demon. This module will introduce a blood-based ex vivo platform that is able to decipher ruminant immune responses against different pathogens. This platform is in line with the 3R principle that fosters replacement, reduction and refinement of animal experimentation. We are going to show you why we developed this platform, how it works, and what kind of data it can generate.
Climate change is real, and we are experiencing that our planet is getting hotter. The picture displays the rise of an average temperature in the last century. Unusual weather events such as heat waves, heavy precipitation, and droughts are on the rise, which affects all living organisms. The changing climate affects also the geographical distribution of vectors such as insects and ticks that can transmit deadly diseases. We have to prepare for incursions of new vectors, including their pathogens, that can affect the productivity of livestock and the provision of animal protein to the people. From this map, we can clearly see 2 different domestication centers of cattle. The Fertile Crescent, where the Bos taurus have been domesticated 10,000 years ago, and the Indian subcontinent, where Bos indicus cattle have been domesticated 8,500 years ago. Let us look at the different visible traits of the cattle. Bos indicus have a fatty hump on their shoulders and a large dewlap, which differentiates them phenotypically from Bos taurus cattle. They are known to be more heat tolerant, probably because they have been domesticated in a hot climate. This hot climate was likely to favor specific pathogens, including vector-borne ones, and therefore domesticated of Bos indicus genetic background are likely to have gone through different selection pressure in terms of pathogens than cattle of a Bos taurus genetic background. Therefore, a different genetic background is likely to result in different immune responses that correlate with different resistance against a pathogen or disease.
Challenge experiments involving the natural host are generally very informative. However, such challenge experiments are difficult to implement since they have to be executed in high security settings that are costly and logistically demanding. Most importantly, they are likely to harm the animals involved in the study. Moreover, to get most meaningful data and enroll only a limited number of animals, such experiments require a robust and reproducible challenge protocol, which is not always at hand or has to be established. Another possibility to investigate disease susceptibility of different breeds is to monitor disease presence in those breeds. This requires the presence of different breeds in the same geographical location so that the infection pressure with a certain pathogen is similar. Unfortunately, such conditions and data are often not available.
As part of a vector and vector-borne disease consortium, we set out to develop a blood-based research platform that allows us to dissect immune responses of different cattle breeds against vector-borne pathogens. Therefore, we took advantage of blood which harbors many different types of immune cells, as can be seen in the illustration. These cells orchestrate the immune response of the native host against the pathogen. Since many vector-borne diseases are transmitted by blood-sucking insects or arachnarids, such as ticks, the immune cells of the blood are clearly important players in the immune response.
So now it gets a bit technical. On the first day, we collect venous blood from several healthy outbred cattle to be used for the analysis. It is of utmost importance that the animals are healthy and represent outbred animals to avoid any bias of the data to be generated. The blood needs to be transported as fast as possible to the laboratory, and peripheral blood mononuclear cells, abbreviated PBMCs, have to be isolated with the pathogen of interest. Therefore, a multiplicity of infection has to be determined, meaning we have to test different ratios of blood cell to pathogen. Then, we infect the PBMC with the pathogen for a defined time and place them in an incubator. The next day, we stain the different immune cells with defined antibodies that recognize specific surface markers of the immune cells that might be expressed in the presence of the pathogen, or that recognize molecules like cytokines. This enables us to monitor the different changes of immune cell populations represented by the PBMC. Additionally, we investigate molecules such as cytokines and chemokines secreted by the different immune cells in the presence or absence of the pathogens using a state-of-the-art bead-based assay. All the data get analyzed using specific software packages such as FlowJo, and this enabled us to characterize the ex vivo immune response of a group of animals belonging to a specific breed. Then we can compare immune responses of different breeds. We investigated the immune responses of both Taurus and both Indicus cattle against the viral arthropod-borne bluetongue virus, abbreviated BTV. Bluetongue virus causes a deadly disease in ruminants characterized by fever, excessive salivation and swelling of the face and tongue, leading to cyanosis and a typical blue tongue. In recent years, European countries faced incursions of the bluetongue virus with many deadly outbreaks. For the analysis, we used blood samples of 8 healthy outbred cattle for each, both Indicus or both Taurus. The most striking finding was the different immune response towards BTV of both Indicus versus both Taurus cattle. While the latter showed a rather over-exuberant response compared to the mainly antiviral-like immune response of both Indicus cattle. The cytokines levels induced by the pathogen exposure and secreted by the immune cells different also significantly. In one farm, where both types of cattle have been kept, the intrusion of the pathogen caused way more sick both Taurus cattle than both Indicus cattle, which supports our hypothesis that different immune responses cause different resistance.
The developed ex vivo platform is a new tool awaiting its use by fellow scientists to screen different breeds for immune responses and to compare the latter. This will help to narrow down resistant breeds that can be further investigated and used for breeding programs. The current different local livestock breeds in Africa, Asia and elsewhere harbor resistance against different pathogens besides other traits, such as productivity and heat tolerance, and we need to protect that diversity for our future. Thank you for your attention.
Thank you.
Hello everyone and welcome. My name is Dennis Getange, a PhD student at Isipe, Kenya, and at the University of KwaZulu-Natal in South Africa. This short module focuses on an innovative technique called direct PCR high-resolution melting analysis, or direct PCR HRM, an exciting molecular diagnostic tool for rapidly detecting tick-borne pathogens directly from samples without DNA extraction. By the end of this module, you will understand what direct PCR HRM is, how it works, and why it is a game changer for disease surveillance, especially here in East Africa. Every year, diseases transmitted by ticks such as anaplasmosis, heartwater, and East Coast fever cost livestock farmers in East Africa tens of millions of dollars and threaten rural livelihoods and food security. Under the Prepare for VBD consortium, we have developed a direct PCR HRM tool that eliminates the need for DNA extraction and could revolutionize regional surveillance of tick-borne pathogens by enabling faster on-site detection. Direct PCR HRM is a technique that eliminates the need for the time-consuming DNA extraction step, thereby allowing us to amplify target DNA directly from a tiny amount of biological material, like a drop of tick hemolymph without DNA extraction. To begin, we need to collect ticks from cattle using a standardized and safe approach. First, approach the animal calmly, ideally with the help of the owner or a handler to restrain it. Then, using blunt forceps, grasp tick as close to the skin as possible and pull it steadily upward. Be careful not to twist, as this could leave the mouthparts behind. Next, place the live ticks into well-labeled microcentrifuge tubes or into well-ventilated collection vials. To keep them alive, you may add a small piece of moist cotton. And one very important reminder, always wear gloves and avoid crushing ticks with your fingers. This helps prevent accidental exposure to pathogens. Once we have live ticks, the next step is to collect hemolymph, the tick's equivalent to blood. Begin by immobilizing the tick on a clean surface using fine forceps. Cut one leg using a sterile scabbard blade. Then gently press the body, taking care not to apply too much pressure. Too much force could rupture the internal organs and release gut contents. Now, using a pipette with a 10 microliter tip, carefully collect the hemolymph. We only need a very small drop, about 1 to 2 microliters. This clear or sometimes slightly reddish fluid should then be placed directly into a sterile microcentrifuge tube. Now that we have our hemolymph sample, here is the simple workflow. First, direct PCR master mix is prepared in the laboratory, and this mixture contains everything we need, that is the polymerase enzyme, buffers, fluorescent dyes, and the primers. And then in the field, the process is very simple. All we need to do is coat the master mix into PCR tubes, and then add the hemolymph sample directly into the mix. Importantly, there is no need for DNA extraction. This makes the process much faster and more efficient. Next is placing the prepared tubes into a portable PCR machine. And the amplification process then begins, generating millions of copies of target DNA. Immediately after the PCR, the HRM analysis begins, where the machine slowly raises temperature while continuously measuring change in fluorescence intensity. As the temperature increases, the double-stranded DNA begins to separate or melt, causing a decrease in fluorescence. Different DNA sequences have different melting temperatures based on their specific nucleotide composition and length. For example, DNA from Anaplasma species will melt at a different temperature than DNA from Ehrlichia species, creating distinct melting profiles. And by comparing these characteristic melting curves or profiles to reference controls, we can determine which pathogens are present in the sample with high precision. The key practical steps in the field are how to safely identify and collect ticks from a host, how to collect a drop of hemolymph from a tick, how to pipette hemolymph into the master mix, and how to run direct PCR-HRM as an on-site diagnostic tool. The key takeaway message: direct PCR-HRM is a simple, scalable, and robust solution for field teams, as it allows for rapid detection of tick-borne pathogens like Ehrlichia and Anaplasma, and significantly reduces costs by eliminating the need for expensive DNA extraction kits. By giving surveillance officers and veterinary professionals the ability to get quick results right where they are, we enable faster, more informed decisions, leading to better targeted interventions, ultimately improving the health of our livestock and protecting communities. Thank you.
Water courses and wet areas in pastures increase liver fluke infection risk. These environments favor intermediate host snails that transmit the infection. Drone aerial surveys map even the smallest water bodies, assessing infection risk across the studied area, including remote locations. This technology provides a powerful tool to evaluate risk across entire grazing areas, helping farmers take targeted action to protect their herds. The diagnosis of Fasciola hepatica mainly relies on copromicroscopical analysis. For accurate results, collect individual rectal fecal samples, minimum 20 grams, from a minimum number of 10 animals per group on the selected farms. Transport the samples to the laboratory on the same day, keeping them cooled below 10 degrees Celsius. If immediate analysis is not possible, store the samples vacuum-packed and refrigerated until testing. The risk of liver fluke infections in pastures can be assessed by collecting intermediate host snails, such as Galba truncatula, from water courses in the studied area and identifying them using both morphological and molecular analysis. The snail species collected on farms can be morphologically identified based on the shape and orientation of their shell aperture. In Europe, the most common intermediate host snail responsible for Fasciola hepatica infections is Galba truncatula, a brown or grayish dextral snail measuring 5 to 10 mm in length. The fluke infection levels in the snails can be assessed by detecting Fasciola hepatica DNA within their tissues. Therefore, we extract DNA from Galba truncatula snails, then perform PCR using species-specific protocols for Fasciola hepatica. The amplified DNA is finally visualized by gel electrophoresis to confirm parasite presence. The University of Naples team has developed a suite of educational resources for this massive open online course. This includes standard operating procedures and instructional videos covering key diagnostic techniques such as mini FlowTAC, fluke finder, and sedimentation for the detection of liver flukes in cattle. An illustrated atlas is also provided featuring images of liver fluke eggs other parasitic elements such as eggs of various helminths and oocysts or cysts of protozoa, as well as common artifacts and pseudoparasites to support accurate identification and interpretation. The improvement of liver fluke diagnosis was achieved through the development of the cubic flow-tac microscope, a digital, compact and portable device designed to analyze fecal samples prepared using mini flow-tac in both laboratory and field settings. Thanks to advanced artificial intelligence software integrated into the cubic flow-tac microscope, fluke egg detection reaches 97% accuracy, enhancing disease monitoring and control in endemic areas. For more details, a recorded webinar on AI-based liver fluke diagnosis, hosted by our University of Naples team, is available online.
This module is on eDNA-based environmental monitoring tools. For snail-borne parasites, here you will be introduced to the concept of environmental DNA, in short called eDNA, and how it can be used as a tool for environmental monitoring of snail-borne parasites, serving as the cost-effective supplement to the traditional snail survey that was introduced earlier in this module. By the end, you'll be able to understand what eDNA is and learn how to perform eDNA-based monitoring of snail-borne parasites in the environment. Environmental DNA, or eDNA, is genetic material shed by organisms into their environment. It can be collected from environmental samples like water, soil, or sediment and analyzed in the laboratory to identify the species present in that location. Here's shown an example with aquatic eDNA, where animals living in water or in close contact with water shed genetic material into the water through, for example, feces, urine, hair, or skin cells, and then eDNA can be detected in water samples that are collected. When it comes to snail-borne parasites and eDNA, here using Fasciola liver flukes as an example, all the parasite stages outside the main host, such as eggs, Myristidia, and Cercaria, all contribute to the pool of parasite eDNA in the water body, and can therefore be detected in water samples. Also, the vector snails, being aquatic or semi-aquatic depending on the species, shed DNA into the same environment and can be detected in the same water sample. There are several advantages of using eDNA methods for snail-borne parasite monitoring and in general. The methods detect DNA traces without finding the target organism itself. Therefore, specialized taxonomical expertise is not necessary for environmental sample collection as it is for snail surveys. And samples can be collected rapidly and also often cover a larger geographical area. During analysis, multiple species, such as both the parasite and snail species, can be detected in the same sample. And lastly, eDNA methods have proven to be highly sensitive when it comes to low abundance target organisms. For snail-borne parasites, that means where there's low transmission and could be either when snails or parasites invade new areas or when parasite control efforts are going towards elimination. Water collection in the field is filtered using filters with very small pore sizes of 0.22 up to 3 micrometers to capture the eDNA. The water filtering can be done either in the laboratory using an electric vacuum pump, but it can also be done in the field immediately after collection. Here a handheld vacuum pump is used attached to a bottle with a funnel and an open filter. The filter with the captured eDNA is then stored either frozen or with a preservation buffer depending on the setting. In the lab, the DNA is extracted from the filters using a standard DNA extraction kit optimized for eDNA. It's important to be careful not to cross-contaminate between the samples. To detect possible eDNA from Fasciola, a PCR is performed using species-specific primers validated for eDNA. If there are bands of the correct length on the gel, Fasciola eDNA has been detected in that specific water sample. Just be aware that currently eDNA methods still cannot distinguish between different life stages of the same species. for example Myricedia and Cercaria. Despite limitations, eDNA holds great untapped potential for environmental monitoring of snail-borne parasites. It enables early detection of low-abundance vector snails, especially as climate and environmental changes shift species distributions. As parasitic diseases in low and middle-income countries move from control to elimination, eDNA offers a sensitive and cost-effective tool for monitoring environmental transmission and complements traditional snail survey methods.